Hello everyone, welcome back to the session. So in the previous classes we have discussed about the carbohydrates, lipids, proteins and amino acids. So we have completed all those things, and in today's class we are going to discuss about enzymes, and in this enzymes will start today and will not going to finish today. We are going to finish this enzyme tomorrow only. So because this enzyme has the most important subtopics under this topic enzyme, we are going to discuss all the information regarding the enzyme only in an exam point of view. Let's start the session. So you should know what is mean by enzyme. So enzyme are biological catalyst that speed up the biochemical reactions, are not consumed in the reaction, work under mild physiological conditions. So these are all the properties of enzyme. So if you want to explain about enzyme, these are all the way we can explain the enzyme. But before all these things, you should know that this enzyme is nothing but a protein, isn't it? So in yesterday's class we have discussed complete information about protein and what are all the substances which are having a functional role. The functional role of protein is one of the functional will be the enzyme. So we can define this enzyme as a biological catalyst. Biological catalyst. So why we are saying this as a catalyst? This is speeding up the reaction. Without enzymes, definitely the reaction is going to happen, but with the presence of enzyme, the reaction will little bit faster. So this is very important. So that is why we are saying this enzyme as a catalyst, biological catalyst. Now it speed up the biochemical reactions. It is speeding up the biological relations. See, just you should know the difference between these things. See, without enzymes, whether the reactants can convert into the product or not. Of course the reactant has the capacity to convert into product, but with the presence of enzyme the reaction will be little bit faster. So this is why we are saying that as a speeding of the biological reactions now are not consumed in the reaction. Whenever any one reactant with the it has to be fused with the reactant 2, in the presence of some enzyme, it has been give one product. So after that, what happened to the enzyme? The reactant, now it is getting converted into a product. Now the enzymes which is responsible for converting this reactant to product, what happened to this enzyme? The enzyme will not consumed in the reaction. Again the reaction is going to help for the another reactant to convert into a product. So you should know that whenever a reaction is taking place with the presence of an with the presence of an enzyme, the enzyme is not getting consumed in the reaction. So work under mild physiological conditions. So under the mild physiological conditions the enzyme will be working on. What is meant by this mild physiological conditions? It may be temperature, it may be pressure, whatever it is, the enzymes need an optimum temperature and pressure. Okay. Now just see, look at this picture. This is an enzyme. So this is an enzyme. Each enzyme always have an active site. Active site in the sense where the substrate, see, this is an enzyme. So in this enzyme we are having an active site where the substrate is going to bind. So the place in the place of an enzyme where the substrate is going to bind, and that place is called as an active site. So whenever the substrate is bind with the active site of an enzyme, that complex is called as the enzyme substrate complex. See, now just listen to this mechanism. This mechanism of action of an enzyme, key and lock hypothesis. See, this is what a lock and key method. For one lock there has to be only one key. Okay, this lock cannot be opened by some other key. So that is what key is a substrate. Here key is the substrate and lock is a particular enzyme. So this enzyme has only one substrate. No other substrate will bind with this lock, that is the enzyme. So the enzyme has, so you should know that there is an enzyme specificity, which means this particular enzyme is bind with these types of substrates only. So the substrate specificity will be seen in this particular lock and key hypothesis. Now, induced fit hypothesis. Just look at this picture. Here, the key is getting correctly fit into the enzyme so that the lock has been opened. Now, just look at this picture. The key here will not fit properly, so that's why there will be no reaction. The lock doesn't open. So, this is what the hypothesis, which is called as a lock and key hypothesis. So, in this hypothesis, we come to know that each enzyme binds with the specific substrate. So that's why we are saying that enzyme specificity is important. Now, what are all the steps? So initially we have an enzyme. We are having different types of an enzyme. Definitely that has to bind with the substrate. So the enzyme will be mentioned by the letter. It's denoted by capital E, and the substrate will be denoted by capital S. So now when the enzyme is bind with the substrate, they are going to form this enzyme substrate complex. Now as a result after the substrate complex has been formed, the product is formed. See, just look at this picture. Enzyme plus substrate gives enzyme substrate complex. Now after the complex has been formed, product will form and the enzyme will be removed. The enzyme is not consumed, enzyme is not changed. Okay, so the enzyme is released unchanged. There will be no changes occur in case of enzyme. So you should know that with the equilibrium of a reaction is not at all affected by the enzyme. The product formation is not at all influenced. The concentration of product will not be influenced by the enzyme. So the only thing what the enzyme will do in a reaction is it is just speeding up the reaction. It is acting as a catalyst. That's all about the enzyme. So this is very important. Now see here, these are all the important properties of an enzyme. Just look at this picture. Enzymes are mostly protein, so that's what we keep on saying, that hormones are nothing but a protein, enzymes are nothing but a protein, and we are having the enzymes which is not a protein, that is what a ribozymes. So this is very important. RNA which is having the catalytic role, that is what we are calling them as a ribozyme, and here it is not called as protein. Right, now see here, highly specific for substrate. This is what we keep on saying, that one enzyme has to be fused with specific substrate, only then the product will be formed. Unless and until the proper substrate is getting attached with the enzyme, there will be no proper product will be formed. The product formation will get affected. So that's what we are saying, highly specific for substrate. So this can be expected in the exam. Enzyme specificity. Now increase reaction rate by lowering the activation energy. This is very important. See, let me say you a two reaction. So here in the reaction 1 we are having a substrate, and this substrate is getting converted into a product. Now in the reaction 2, now enzyme plus substrate together, they are forming an enzyme substrate complex, and as a result enzyme will be released unchanged and the product will be formed. Now, so here also the substrate is getting converted into a product. Here also a substrate is getting converted into a product. But what will be the difference between the reaction 1 and reaction 2? Here there will be no involvement of enzyme. Here there will be an involvement of enzyme. So what is with respect to this activation energy? Just look at this reaction 1. Whenever the energy invested in this reaction, the energy activated to convert the substrate to the product is very high. With the presence of this enzyme, the activation energy is very low. So this is very important. This can be expected in a question. Do not change equilibrium constant. So with the enzyme will not change anything in the reaction. Just the only function of the reaction will only function of the enzyme will be to speed up, to enhance the reaction. So this is very important property. Now comes to the nature and composition of an enzyme. So what about this nature and composition of an enzyme? So protein enzymes, most enzymes Just look at this point, this is very important. Most enzymes or globular proteins have a specific 3D structure essential for function. See, this protein enzymes, most enzymes are globular proteins. So in yesterday's class we have discussed, based on the shape we are having two different types of proteins. One is globular protein and another one is fibrous protein. So here now we are discussing about an enzyme. So these are all the protein enzymes. The protein enzymes are usually globular in shape. So that's why we are coining the term as globular protein. Have a specific 3D structure which is essential for function, and this is very important. So as we have discussed in yesterday's class, we are having three different types of, we are having many types of many structure of protein. There is primary structure, secondary, tertiary and quaternary structure, no doubt about that. But which structure gives a 3D dimensional view of a protein? And that is nothing but a tertiary structure. In tertiary structure we are having a three-dimensional view, three-dimensional structure of a protein. And why we have a three-dimensional structure? Because that structure is the one essential for a proper function. So you should know this protein enzymes which are globular proteins, as it is globular in shape, and they have a specific 3D structure, and this is essential for the function. And what are all the examples for the globular proteins and which are having 3D structure? That is amylase, protease and DNA polymerase. All these are very important protein enzymes. Now, ribozymes. So just now we have studied all enzymes are proteins except the ribosome. So what is ribosomes then? So the ribosomes is the RNA enzyme. So in the previous slide we have studied about the protein enzyme and what will be the properties of the protein enzyme and some of the examples. Now look at this picture, look at this slide. Ribosome is nothing but this RNA enzyme. So this RNA molecules, they are having a catalytic activity. So this catalytic activity, those are having this catalytic activity which had by this RNA molecule. So that's why we are saying this RNA enzyme as ribozyme. And what are all the examples of this ribozyme? rRNA, that is ribosomal RNA, peptidyl transferase activity and self-splicing intron. So all these were the example for the ribozyme, isn't it? So now we are having a different components of an enzyme. We are going to discuss about that. Just to see, look at this picture. Apoenzyme, we are having a cofactor and together we are calling them as a holoenzyme. So as of now, just remember that the enzyme apoenzyme plus cofactor, together we are having a holoenzyme. So holo in the sense complete. So the enzyme which is complete only when this apoenzyme is combined with the cofactor. When the apoenzyme is combined with the cofactor, only then we are calling that particular structure as a complete enzyme, that is the holoenzyme. So why the cofactor is essential for a holoenzyme? One, what is apoenzyme? What is the function of this cofactor? Everything will be discussed in the upcoming slides. So all these things what we have discussed as apoenzyme, cofactor, holoenzyme. So these are all were the components of an enzyme. Just look at this slide. Many enzymes are not functional alone. Many enzymes are not functional alone. They are having certain other things also. Say for an example, apoenzyme, protein part of the enzyme. So this is very important. So you should know that. So in yesterday's class we have discussed glycoproteins, lipoproteins. So the non-protein part, that is what a prosthetic group. So here in glycoprotein, the non-protein part will be the glyco and the protein part is present here, and this non-protein part is called as a prosthetic group. So like in the same way, this enzyme is also having the non-protein part as well as the protein part. So now in the enzyme, we have studied this enzyme as a proteinaceous in nature, no doubt about that, nothing wrong in that. And now in this enzyme, They are also having a non-protein part and they are also having a protein part. So that's what apoenzyme, this is what a protein part of an enzyme, and this apoenzyme, when it is not combined with any of the cofactor, we are calling this apoenzyme as inactive. When it is alone, when apoenzyme is alone, when it is not combined with any of the cofactor, we are calling this particular apoenzyme as inactive. It is inactive in nature, but this apoenzyme is a protein part of the enzyme. Now what about this cofactor? So this cofactor is a non-protein part required for activity. This is very important. See, we have studied this apoenzyme. So this apoenzyme, they are protein part of an enzyme and it is inactive. If apoenzyme has to be activated, it needs cofactor, and cofactor is a non-proteinaceous in nature and it is required for the activation of this apoenzyme. When the apoenzyme is combined with the cofactor, only then we can call that particular term as a holoenzyme. Now what are all the things which are acting as a cofactor? We are having a metal ions like magnesium, zinc, iron, copper. So all these ions are called as a cofactor, isn't it? Now organic molecules are coenzymes, and again this coenzyme, it is also called as a cofactor. So this is very important. Just look at this picture. Minerals as enzyme cofactor. Most of an enzyme need mineral as a cofactor for activating them. Just look at this picture. This is an enzyme, and this enzyme having some site where the substrate is getting attached with it, and that is called as an active site, and in the active site the substrate is going to bind. Just look at this picture. This enzyme is not functioning. Why? Because with the presence of the cofactor, say for an example any mineral ion, only with the presence of this mineral ion, this enzyme function is getting activated. So you should know, for the activation of an enzyme, we definitely need a cofactor. Very important. Say for an example, iron for heme production. We are having a hemoglobin, no doubt in that, and the hemoglobin we are having a heme group and globin group. Globin is nothing but a protein, and the heme, for the heme production we need the central metal atom will be the ion. So ion here is acting as a cofactor for the heme production. Now for DNA synthesis, magnesium and zinc plays a crucial role in the DNA synthesis. So if any product has to form, we need an enzyme, and if an enzyme has to be activated, it needs a cofactor. So all these points are very important. Now holoenzyme, as we said said in the previous slides, this holoenzyme is an active enzyme, and this holoenzyme, whenever the apoenzyme is mixed with the cofactor, we are saying this particular thing as a holoenzyme. Now just look at this picture. You can understand very clearly. This is an apoenzyme. With the presence of this cofactor or a coenzyme, it first fixes with the active site of an enzyme, and after that the substrate is getting attached with the whole so now see here, when the apoenzyme is bind with a cofactor or any coenzyme, we are calling this thing as a holoenzyme. Now only then this holoenzyme is mixed with the substrate, we are saying this complex as an enzyme substrate complex, isn't it? Now just types of a cofactor. We are having a different types of cofactor. That is, a coenzymes, organic molecules are loosely bound. See here, let me explain you one by one. We are having a different types of cofactor, and the first type of cofactor will be the coenzyme. So in this coenzyme, we are having an organic molecules. See, organic molecules are acting as a coenzyme, and we need, as I mentioned you before, if the apoenzyme has to be active, if the apoenzyme wants to active, definitely it needs a cofactor or a coenzyme. Either it needs a cofactor or a coenzyme. And if the coenzyme wants to, what this coenzyme, okay, what this coenzyme? This coenzyme is nothing but an organic molecules. So here the coenzyme is acting as a cofactor. Now these coenzymes are nothing but an organic molecules. It is always associated with an coenzyme, not tightly but loosely. So that's why we are saying loosely bound. This is very important question. Now, what are all the examples which are acting as a coenzyme? That is NAD plus, FAD, that coenzyme A. So, all these are very important. So, kindly make a note of it. This can be expected in our exam. Now, prosthetic groups. Now, this prosthetic groups is again another type of cofactor. It is tightly bound with the coenzyme. Okay. So, it is tightly bound with the coenzyme. Example, the heme group, which is associated with the ion. Biotin, all these is considered as a prosthetic group. Now, metal activators. Some of the metals are also very important for the activation of the apoenzyme. And what are all those? Magnesium for kinases, for all the kinase activity, kinase enzyme, which means ATP adding enzymes. That was kinase activity. So all those enzymes, definitely they need a magnesium. Now zinc is essential for the carbonic anhydrase. Carbonic anhydrase is very important in maintaining the buffer in the body. Now, so this is very important. So in this slide we are going to discuss about the classification of an enzyme. So the enzymes have six different class, right? So we are having six different classes of an enzyme. So first type will be the oxidoreductases. See, oxido, it is also important for doing oxidation, and it is also important for the reduction process. So if the enzyme, if it is doing oxidation or reduction, if it is involving in oxidation and reduction activity, we are categorized that particular enzyme under the oxidoreductase. The example for this oxidoreductase will be the dehydrogenase. And the second type will be the transferase enzyme. So this transferase enzyme, it is nothing but a kinase enzyme. Those enzyme which is having a suffix as kinase, all those were coming under the transferases. Why? Because it is transferring the phosphate group. So hydrolases, example protease enzyme, everything is categorized under the hydrolases. And lyases means decarboxylase. Those enzymes which are involving in removing the carbon. So we are saying that as carbon dioxide. So we are categorized them under the lyase group. Now isomerase is the racemase. Racemase is an enzyme which is categorized them under the isomerases. And ligases is also called as the synthetases. Synthetases is an enzyme, it is also called as the ligases. The best example will be the DNA ligase. So all these are the important classification of an enzyme. Now, how we are naming the enzyme? So enzyme nomenclature are very important. So how can we name the particular enzyme? Usually the name has 3 words. That is what trivial names. Example, pepsin, trypsin, and we are also having a systemic names based on the structure and function. See here, so how all these nomenclature has been prescribed based on substrate and what reaction they are involving in. So based on all these things, we are naming the enzymes. So the best example will be the lactate dehydrogenase. So you should know that particular enzyme which is going to bind with the lactose. So if lactose is a substrate and it is involving in the dehydrogenation reaction, we are naming that particular enzyme as lactate dehydrogenase enzyme. Now see here, enzyme commission number. So see, we are having an EC number. EC usually represent for enzyme commission. So the what will be the format? ECXXXX. Usually they are having a 4 numbers. So the best example will be the alcohol dehydrogenase election commission. Enzyme commission 1, 2, 3, 4. So these are all the numbers which we are attached with the enzymes. Now, so the active site of an enzyme. So the active site of an enzyme, usually they are having a feature. Just now I have explained you in the picture. Consider this as an enzyme, and the site where the substrate is getting attached, this site is called as an active site. So this site is called as a small pocket, or we can call them as a cleft. Now it is a specific for substrate. So usually those substrate which are having like in this structure, it can be easily bind with this enzyme. So if they are having this, if the substrate is in this shape, whether it has whether it can able to bind with this enzyme? No. So that's what specific for certain substrates. Now, what this active sites exactly composed of? They contain amino acids that participates in the catalysis reaction. Again, this enzyme that is in the active site, they are having an amino acids that participate in the catalysis reaction. So this is very important, and usually the enzyme have, in the active sites they are having a 2 regions, binding site and the catalytic site. One is essential for attaching with the substrate, and one site is meant for the functional role, that is the catalysis role. See the same thing, this is the substrate and this is an enzyme which have an active site. Now the enzyme and substrate is getting bind with each other, so we are calling this complex as the enzyme substrate complex. Now see again the same thing in the protein. See, this is the scaffold. Why the protein in like this structure? Scaffold protein. Scaffold to support and position of an active site. So in this active site of a protein, we are having a binding site. The blue color region, all these are called the binding site. So in the active sites itself, we are having a 2 regions. One is the binding site and another one is the catalytic site. Isn't it? Now in this binding site, we are having a bind and orient the substrates. Bind and orient with the substrate. The catalytic role, they reduce the chemical and activation energy. So whenever we are studying about the activation energy, enzymes always lowering the activation energy. Without the enzyme, the reaction, the activation energy of the particular reaction will be very higher. With the presence of an enzyme, the activation energy will be lower and the product formation will be earlier. So this is very, very important. Now, model of an enzyme action. So we are having 2 different types of model. One is lock and key model and another model will be the induced fit model. So lock and key model is proposed by M.L. Fisher. So this lock and key model I have explained you in the starting slide itself. So we are having one sub enzyme. So in one enzyme we are having so if this is an enzyme, if this is a substrate, if this substrate can be easily bind with this enzyme. What if the substrate will be in this shape? What if the substrate is in this shape? This cannot be bind with this enzyme. So that is what, for one key there is only one lock. If the key is not proper for the lock, it cannot be unlocked. So this is what a lock and key model proposed by the M.L. Fisher. And here the substrate fits exactly into the enzyme. So this is what an enzyme. It is mixed with the substrate. So together we are calling them as an enzyme substrate complex and enzyme, and now the product has been formed. So this is important. Now induced fit model. Now this model has been proposed by Koshland. What he is saying against this lock and key model? Again, he is not accepting the lock and key model. Why? Because the enzyme will keep on change according to the structure of a substrate. So that's what the theory of this Koshland. And when compared to this lock and key model and the induced fit model will be more accurate. Okay, it is flexible, but this lock and key model is very rigid. and this is the most accurate model, and this is very important. And what is meant by induced fit model? Once again, the enzyme will change its shape according to the shape of the substrate. So this is what an induced fit model. Now just look at this picture. So this is an enzyme and this is a substrate. Now the enzyme is fixed with the substrate properly, so this model is called as a lock and key model. Now just look at this induced fit model. So this is an enzyme, and here it is a substrate. Now here, see here, even though the active site are in different shape, the substrate is in circular, and it will not fit with the enzyme. But just see here, after the binding, the enzyme has been changing its shape. Together they are forming the enzyme substrate complex. So this is what an induced fit model and the lock and key model. And the most accurate model will be the induced fit model. Now, enzyme specificity. So usually we are having 4 different types of specificity under the enzyme, and the first specificity will be the absolute specificity. This enzyme will be bind with only this substrate. So this is what absolute specificity. Say for an example, urease is an enzyme. It acts only on the urea. Here urease is an enzyme and the urea will be the substrate. And this enzyme will always acts only on this substrate called urea, and none of the other substrate will be done by this urease. So this is the example for the absolute specificity. Now group specificity, as the name suggests, they are acting on similar functional groups. Here it is not one enzyme, one substrate. So those enzyme, see here, many, the hexokinase is an enzyme. Hexokinase, in the sense, those enzyme which ends with the word kinase, they are involving in the phosphorylation activity. Phosphorylation activity in the sense adding a phosphate group. So hexokinase phosphorylates many hexoses. Hexoses means 6 carbon containing sugars, carbohydrates. So it's 6 carbon containing compounds, not only the sugar. So in all these hexoses, this hexokinase will phosphorylating. So acts on the similar functional groups. So similar functional groups, this enzyme can work on it. Okay. So that's what a group specificity. Now the third important thing will be the bond specificity. What is this bond specificity? This acts on specific bond only. Say for an example, peptide bonds. So peptide bonds, you all familiar with the word peptide bonds, peptide, polypeptide and proteins, amino acids and all those things. I don't want to dig deep into that because we have discussed very clearly all these things. Now see here, pepsin again is an enzyme, breaks the peptide bonds. So this pepsin will not going to break the new glycosidic bond. This pepsin will not going to bind, going to break a hydrogen bond. So here the pepsin, wherever it is seeing the peptide bond, they are going to break the peptide bond. So that's what a pepsin, they are having an activity only on the specific bonds, specifically the peptide bond. Now just look at this picture, stereo specificity, specific for optical isomers, and L amino acid oxidase. So this optical isomers means enantiomer, diastereomer, and those they are having a dextrose or levorotatory. So all these things, so those we are having a under the plane polarized light, they are having any rotation or not. So that in any enzyme which is acting on that specificity, that is called as a stereo specificity, and the example will be the L amino acid oxidase. Now, what are all the mechanism of an enzyme action? What are all the steps which are involving in the enzyme action? Just anyhow, they are going to, the enzyme has to bind with the substrate. Now after the binding, it forms an enzyme substrate complex. Now here the transition state stabilized. Now the product will be formed as enzyme plus product. This is what the mechanism of an enzyme. Here the enzyme remains unchanged. This is an important point. And activation energy of an enzyme, minimum energy needed to start a reaction. The enzyme doesn't need more energy. The enzyme needs a little bit of energy. A very minimal amount of energy is sufficient to start a reaction. So that's why we are saying enzymes have enzyme lowering the activation energy. And if the enzyme is not present, the reaction having a higher activation energy. And another important thing you should know, the enzymes, they are not affecting the Gibbs free energy. They are not affecting the equilibrium. So all these are very important. This can be expected. So now, as I have mentioned you before, these are all the classes of an enzymes and their reaction, what these enzymes are involving in, and the examples for all these things. Kindly make a note of it. All these are very, very important. Now, so we are going to start about an enzyme kinetics in tomorrow's class. Okay, so just to go through all these things, we'll meet you in the next class. Thank you so much.